Hello, dear students. Welcome to my channel. It's me, Bushra Aman, and here we'll study 12th Standard Biology. Part 4, Reproduction in Higher and Lower Plants. Points of this video. The first one is the pollen pistil interaction, artificial interaction, double fertilization, development of embryo, and the last one is the development of endosperm. Here in this video, we'll cover all these 5 points. Pollen pistil interaction in a dynamic continuous process involving communication and selection between the pollen grains and a female reproductive parts of a flower like pistil, which ultimately determines whether the fertilization will occur or not. This interaction is crucial for ensuring that a plant is fertilized by pollen from its own species and to prevent self pollination in many plants, thus promoting genetic diversity. Now, here we'll see the key stages of pollen pistil interaction process. The interaction unfolds in a series of steps. Okay, so first, as you can see in the figure, all the steps are mentioned here, but we'll see the step by step. See, the first one is a pollen adhesion and a recognition. Means the pollen grain will adhesion will takes place of pollen grain. Now what is mean by adhesion? Adhesion means to stick, the sticking of the pollen grain, and the recognition by whom? By the pistil. Whose recognition will takes place here? The recognition of pollen grain will takes place. Okay, so see here, the process begins when a pollen grain lands on the stigma. Okay, the pollen grain will, first of all, the pollen grain will come through the wind or through the water by any other sources. Okay, and then what? As you all know, the stigma is a sticky part. The pollen grain will get stick on the stigma. Okay, the sticky receptive tip of a pistil. Okay, the stigma has a special ability to recognize if a pollen grain is, so, the stigma has a special ability to recognize the specific pollen grains which is compatible or not from the same species. Means if it will be from the different species, it will get rejected, or incompatible from the different species or from the same plant in a self incompatible species, which means the stigma is having the ability to recognize the pollinated pollen grain. Okay, so this recognition is biochemical dialogue involving proteins and other molecules on the surface of the both pollen and the stigma. If the pollen is recognized as incompatible, the pistil rejects it, preventing its germination. Okay, so the students, as you can see in the figure, the first step is the pollination. Second step, pollen adhesion, means the sticking of pollen grain. Third, pollen hydration. The nourishment will provide to the pollen to get hydrated. Then the next step is the pollen germination. The process of germination takes place here if it will get recognized. The next step is the pollen tube entry into the stigma. The pollen grain will get, if the pollen grain has get recognized, then it will get entry into the stigma. Now the next step is by moving through the pollen tube, the pollen grain will enter in the stigma. And the next step is the pollen tube growth through the style. Okay, So the pollen tube will move downwards and it will go through the style. The next step is the pollen tube entry into the ovule and embryo sac. Okay, and the next is the double fertilization takes place. Finally, here the double fertilization process will takes place. Now here we'll see the further steps of this process. Okay, so the step is the pollen hydration and germination. As we have studied in the previous slides, the step by step process. Now we'll elaborate it. Okay, so the pollen hydration and germination. Now we'll see what will happen in this step. If the pollen is compatible, the stigma provides it with the moisture and nutrients it needs to hydrate. Okay. Here the hydration of pollen grains according to their need will takes place. This hydration triggers the pollen grain to germinate. Okay, if the pollen is not having the sufficient hydration or the nutrients, it will that that gap will fill up here. Okay, a thin double like structure called a pollen tube begin to emerge from one of the pollen grains spore. Okay, so the pollen tube will emerge here. Okay, now the structure of what is the structure of the pollen tube is it is a thin and doubled layer. A sac like structure will emerge here. Okay, that is called as the pollen tube. Now the next step is the pollen tube growth. The growth of pollen tubes will take place in this step. Okay, the pollen tube then grows through the tissue of the style, the stalk connecting the stigma to the ovary. This growth is not random. It is it is guided by the complex set of chemical signals which is produced by the pistil, a process known as chemotropism. Okay, this process is called as chemotropism, which process the pollen tube formation process. The pollen tube act as a delivery system of a male gametes, that is sperm cells, navigating it navigating its way through the style towards the ovule in the ovary. Okay, now the next step is the entry into the ovule and ovule fertilization. Now this is a final step. The pollen grain will get nourished and it will get hydrated, and the pollen tube will has been formed, and now the ovule formation will takes place. The pollen will get entry finally into the stigma, and here that pollen grain will turns into the ovule and the fertilization process will complete over here. Okay, now see, we'll see the overlook. Once a pollen tube reaches the ovary, it enters the ovule, typically through a small opening called micropyle. Inside the ovule, the pollen tube releases the two sperm cells it carries. One sperm cell fuses with the egg cell to form the zygote, which will develop into the plant embryo. The another sperm the another sperm fuses with the two polar nuclei to form the endosperm, the nutrient-rich tissues that nourishes the developing embryo. This double fusion is known as the double fertilization, a hallmark of flowering plant. Artificial hybridization, as you can see in the figure, it is controlled. It is a process, the process of hybridization, which is takes place artificially. Okay, or in a controlled condition. It is controlled plant breeding technique used to create new plant varieties with desired characteristics. This process involves the intentional cross pollination of two genetically different parent plants to produce a hybrid offspring that combines the superior traits of both. It is a key method in agriculture and horticulture to develop cross crops with improved yield, diseases resistant, and adaptability. So, in natural hybridization, the conditions will not in and control, but that's why the some crops will yield with some diseases, with some inherent defects. Okay, so that's why the human beings or some farmers or some scientists, they will perform this process. They will control the conditions in an lab grown crops. Key steps of artificial hybridization. Now here we'll see the whole process of hybridization process. Okay, the first step is the selection of parent. Okay, now, what is mean by here is the selection of the parent crops, desired parent crops. The first step is to identify two parent plants with complementary desirable traits. Whichever traits we will required, we have to select those parents. Okay. For example, on plant might have a high yield. Some plants have high yield while the other is resistant to a specific diseases. So we have to choose the desirable plant. Okay. Now the second one is emasculation, the crucial step. This is very crucial step which involves the removing the male reproductive part, means the anther, from the flower of a female parent plant before the pollen is released. Okay, before the releasing of pollen grains, we have to remove the pollen, we have to remove the anther from the female plant. Okay, this is done to prevent self pollination. We don't want self pollination here. We have to manage everything in a controlled condition and ensure that only the desired pollen is used for the fertilization. Whatever the suitable trait having that pollen, we have to fertilize that pollen only. This step is not necessary for the unisexual or the female flowers. Now, the next step is the bagging. Now, after emasculation, the flower of a female parent covered with a bag. Okay, the parent plants which will covered with a sack or a bag, usually made up of butter, paper, or cloth, to protect it from contamination by unwanted pollen from external source like wind or an insect. So, as you can know, the crops yield in an open condition, open farms, open fields. So, to prevent the unwanted pollination, that parent plant will covered by the cloth or by some other butter paper sack. Now, the next step is the pollination process. Once the stigma of a bagged female flower becomes receptive, pollen from the selected male parent is manually collected and dusted into it. The next step, rebagging. The pollinated flower is immediately recovered with the bag to protect it from any further contamination. The bag is tagged with detail about the parent plant and the date of pollination. Now, the next step is seed collection. After successful fertilization, the flower develops into a fruit or a seed pod. The seed from this hybrid are then collected, planted, and evaluated for the desired traits. Artificial hybridization is a systematic process that ensures controlled cross pollination, leading to the development of new plant varieties with improved traits. Double fertilization. It is a complex and unique fertilization process found exclusively in flowering plants called angiosperms. It involves two separate fusion events within the female gametophyte, embryos or the embryo sac, leading to the formation of both the embryo and endosperm in a seeds. Double fertilization is significant in plants because it ensures the coordinated formation of both a diploid embryo and a triploid endosperm, which is crucial for the survival and nourishment of the developing plant. This unique process is a hallmark of flowering plant called angiosperms and gives them a significant evolutionary advantage over other plant groups like gymnosperms. The key points of significance. The first one is the nourishment for the embryo. The most vital role of double fertilization is the formation of the endosperm, the nutrient rich tissue. The endosperm provides essential food in the form of starch, protein, and fats to the developing embryo. This ensures the embryo has a reliable food source for the growth and germination, especially before it can perform photosynthesis. Like- Now, the next one is the efficient energy uses. In angiosperms, the endosperm is only formed after the egg cell has been successfully fertilized. This is highly efficient because the plant doesn't waste energy creating a good food. It reserves unless a viable embryo has been secured. In contrast, in gymnosperm, the nutritive tissues forms before fertilization, which can be waste of resources if fertilization fails. Now the next one is the stimulation seeds and fruit development. The double fertilization triggers the ovary to develop into a fruit and the ovules to develop into seeds. The hormones released during the process stimulate these post fertilization changes, ensuring the protection and successful dispersal of the seed. Now, the last one is the restoration of diplody. The fusion of the haploid male gamete with the haploid egg cell restores the diploid, which is 2 in number con- condition, which is essential for the formation of the zygote and the subsequent growth of a new diploid sporophyte, the main plant body. The development of endosperm. It is the post fertilization event in flowering plant called angiosperms that provides nourishment to the developing embryo. It begins with the primary endosperm nucleus, a triploid which is 3 in number, cell formed during double fertilization. The PEN means primary endosperm nucleus undergoes repeated mitotic divisions, but the the manner in which it develops into the endosperm tissues varies among plant species. Types of endosperm development. There are three primary types of endosperm development classified based on the pattern of cell wall formation. The first one is a nuclear endosperm. This is the most common types. The primary endonucleus undergoes a series of free nuclear division without the formation of cell wall called cytokinesis. This results in the multinucleated cytoplasm. A large central vacuole often forms pushing the nuclei to the primary periphery. Later, cell walls may form, making tissue cellular. A classic example is the coconut, where the liquid coconut water is a free nuclear endosperm and the solid coconut meat is a cellular endosperm. Now, endosperm. In this type, every nuclear division of primary endonucleus is followed immedi- immediately by cytokinesis and the formation of the cell wall. This results in a completely cellular endosperm from the very beginning. Examples include petunia and Dhatura. Now the next one is Halobial endosperm. It is an intermediate type combining features of both nuclear and cellular development. The first division of the primary endonucleus is followed by a cell wall, dividing the embryo sac into a large micropylar chamber and a smaller chamber may undergoes a few division or none at all. This type is common in many monocots. Now the next one is the ultimately the function of the endosperm. It is to serve as a food reserve for the growing embryo. In some plants like peas and beans, the endosperm is completely absorbed by the developing embryo and the food is stored in the cotyledons. These seeds are called non endospermic or exalbuminous. In other plants, such as maize, wheat, and castor, the endosperm persists in the mature seeds and provides nourishment during germination. These seeds are called endospermic or albuminous. The endosperm plays a crucial role. It provides nourishment to the developing embryo. This endosperm formation type of endosperm provides a detailed explanation of the different types of endosperm development. Now, based on their development process, there are three primary type of endosperms in a plant. The first is nuclear, second one cellular, and the third one is halobial. These three types begins with the division of triploid primary endosperm nucleus, but they differ in how and when cell wall forms. The first one is nuclear endosperm. This is the most common type. The primary endonucleus undergoes a series of free nuclear division, meaning the nucleus divides but the cytoplasm close not does not. So, no cell wall are formed initially. This results in a multinucleated cell. A large central nucleus often develops, pushing the nuclei to the periphery. Later, cell wall may form, making the tissue cellular. Now, here are the example. Coconut is a great example. The coconut water is a free nuclear endosperm, while the solid white meat is a cellular endosperm that forms later. Other examples include maize, wheat, and sunflower. Now the second one is a cellular endosperm. In this type, every nuclear division is immediately followed by the formation of cell wall. The endosperm is nucleus. Is a cellular form. The very beginning. There is no free nuclear stage. Examples: plant like petunia and datura exhibit this type of endosperm development. Now see here the hylobial endosperm. This is a combination of nuclear and cellular type. The first division of the PEN is followed by a cell wall, dividing the embryo sac into a larger micropolar chamber and a smaller chisel chamber. The nucleus in the large micropyle chamber then undergoes a series of free nuclear division, smaller to a nucleus type. The nucleus in a smaller chisel chamber may divide or not, but eventually undergoes degenerators. The development of plant embryo. It is a process known as embryogenesis. It begins with the egg cell zygote and ends with a mature dormant embryo and seeds. This entire process is crucial for establishing a basic body plan of the future plant. The stages of dicot embryo development. The first stage is pro-embryo stage, and the second one is three lobular stage. Okay, the embryogenesis of a typical dicot plant like pea or bean is sequential process characterized by distinct morphological stages. Okay, as you can see in a figure, the all the step by step stages will show. Monocot embryo development. Monocot embryo development is similar in its initial stages but differs significantly in a mature structure. The key difference is the presence of only one cotyledon called saculum. The monocot embryo development. The monocot embryo development is similar in its initial stages, but it is different in the nature structure. A key difference is the presence of only one cotyledon called skeleton. The skeleton is shield shaped and located literally on the embryonal axis. I hope you like this video. So please subscribe, share, and watch the full video without forwarding it. Thanks for watching. In the next video, we'll cover the following points like seed and fruit development, bean seed like dicot, and the maize seed that is monocot, and next one, we will cover the significance of seeds and fruit formation and seed dormancy and apo- apomixis, recurrent apomixis, non-recurrent apomixis, and addentive embryony. The next is the parthenocarpy, polyembryony, and we will cover the cleavage polyembryony, addentive polyembryony. So, in the next video, we'll cover all such points and we will end this chapter in the next video. So, for this chapter 1, Total 5 parts are available